Hello, young people. Today we are going to be learning about DNA. Now, what is DNA? Well, I know you've heard of it. We've talked about it. It is what makes you, you. Now, every living thing on the entire planet has DNA. If you're alive, you've got DNA. But we know that eukaryotes, such as plants and animals like us, have our DNA inside of a nucleus. Now, what is DNA? Yes, it makes you who you are, but it is actually two different backbones that are connected by base pairs. Well, a pair is two, right? So there are two sections of base pairs. A goes with T, C goes with G. So think CG, AT. I always like to remember AT and T. So we have those base pairs and they always match up. And that A goes with the T, the C goes with the G, and over and over and over and over and over again, so microscopically that you have over 9 feet of DNA in your body. Well, Miss Humphreys, isn't my small intestine like 20 feet? That's no big deal. Your small intestine's about this big. Your DNA is about that big. So there's over 9 feet of DNA. That's so much coding. Can something go wrong? Absolutely. If something goes wrong in the coding of your DNA, it's called a mutation. This can be everything from different eye colors, patch of gray in your hair, to a random freckle. That is all coding in your DNA that could have possibly gone wrong somewhere. Not a problem. Most of the time, we just move on perfectly fine without noticing it. Well, Ms. Humphries, you said there's 9 feet. Was just free floating in my body in these nucleuses? Well, we know that our DNA compresses, gets really tight, and it makes these things called chromosomes. Chromosomes. The average person has 46. It's completely okay to have 47. It's just how you get Down syndrome, makes you think a little bit different. So we have the average, again, person, has 46 chromosomes. And inside of those chromosomes are genes that we've learned about. And we know we get our genes from our mom and our dad, and those wind up and create us. So what we're going to do today is actually pull that DNA out of the nucleus of a eukaryote. So today we are working with strawberries. Now you absolutely can extract your own DNA. However, it's gross. It's COVID. I don't want your spit or your respiratory droplets. You don't want mine. And also Ms. Humphries hates spit, so we're not doing it that way. And unfortunately, again, because of COVID, we can't share supplies, and there's quite a few supplies to this. So we're stuck on a demonstration again, but I hope this will suffice. And we will begin breaking down our barriers on the outside of our nucleus. So we have to think about what's in the way of our DNA. Well, we know DNA is inside of a nucleus, so we have to break through that. And we know that all cells have a cell membrane, so we have to break through that too. This is so much work. Let's get started. The very first piece we need to break through is the strawberry itself. So we are going to take our strawberry and smoosh it. Smoosh it. Similar to mechanical digestion, we're going to smoosh it into our bag, breaking down those cells, kind of tearing them apart so we can get some individual organelles out here, like the nucleus and DNA. So I'm going to throw this into my bag, and I shall begin smooshing. And all this is doing is just kind of beginning our process. This is allowing us to get really into those cell membranes, into that DNA, into that nucleus, into that nuclear membrane. We're breaking through the cytoplasm, getting all those good organelles we learned about. We're going to break through all of those. We want kind of a strawberry puree or strawberry juice to start with. So we're going to make sure we get all the chunks. All the chunks. All right. Once we have kind of a chunky strawberry puree, what we now need to do is break through both that cell membrane and that nuclear membrane to get through the membranes. Well, something that you didn't learn because you're only in 7th grade is that membranes have a fatty outside. It's got kind of an oily outside. So what we want to do is break through that oil with a degreaser. Now, dishwasher detergent is very helpful in breaking down fats and grease, and that's why rescue workers use it on animals who are stuck in oil spills, is because it really breaks through that fat. So what we're going to do is add a little bit of dishwasher detergent to break through that fatty cell membrane. Then we're going to just throw a little bit of salt in here because that's going to allow that membrane to separate. We're going to add a little bit of coarseness, a little granulation, and really break apart that cell. So I have soapy, salty strawberry mush. Yum. Who wants it? So good. And we're going to mix all of that together again so we get kind of this broken apart cellular mush. Broken apart cellular mush. Now I have all of these chunky bits, and all I want is the actual liquid out of here. So we've broken through the cell wall, or excuse me, yeah, it's a plant, it's a cell wall, cell membrane, and hopefully the nucleus. So now we are going to separate our mixture, separate the chunky parts of the plant from its DNA. So we're going to take our mixture, and we're just using a good old-fashioned coffee filter. We used to use these before they were Keurigs, guys. You'd make like a whole pot instead of just 1 cup. It's wild. So we're just going to let that filter right on through, and we should get a nice liquid that comes out of that bottom. So while this is processing, let's go ahead and remember a few of our vocabulary terms. We know that we just learned that we have 46 chromosomes in our body that are made up of wound up DNA. Well, that DNA is made up of sections called genes that code for specific traits. So mom gave me 1 allele, dad gave me 1 allele, and remember mom and dad is just an egg and a sperm, each gave me an allele, and that combined to make me. Well, they only donated 1, so there's a dominant and a recessive, and that dominant is always going to show. And that's how you get your traits. That's how you end up looking the way you do. So what we've done now is separate that liquid from the components itself, and hopefully all we have left in here is some DNA. Well, DNA is, like we said, a large matrix that is wound up on each other. So what we need to do is pull it out of its substances. And DNA dissolves in water, but not alcohol. So what we're going to do is take ice cold rubbing alcohol, and we're going to put just a little bit in here so that the DNA does not dissolve. This alcohol will allow the DNA to separate out because, again, it's not going to dissolve in that water. Put a little bit of alcohol in here, swish that around, and allow that DNA to separate from the rest of the mixture. What we should see is kind of a white, stringy substance coming out. And all we're going to do is just grab that with our, let me see if y'all can see that. This is horrible camera work. I'm so sorry, y'all. So what we're going to do is pull this white stringy out of the cell. Look at all this DNA we have. That's an enormous amount of DNA. Look at all that DNA. So I'm going to grab this. I'm going to place it onto my slide so that I can see what DNA looks like under a microscope. And here's the tricky part, getting it off. So I'm going to put it on, place my cover slip over it, and simply slide my toothpick out. All right, let's check it under the microscope.